right, good morning, everyone. If we could regather, and anybody out there who wants to come on in and sit down for this part, we get to be together as long as you want to be together, or until 12:45, whichever comes first. At 12:45, I run and go catch the Uber to go to O'Hare. So, but if we use all of that time, I'll love it. If you're done with me, then you're done with me. And, but this is our time together to do SADS Live, episode 155, in person. So, but before we do that, we're handing out some cards. And if you get these, this is, some of you have heard, well, while we're doing this, just out of curiosity, just in case we're not sure, I'm Mike Ackerman. I live in Rochester, Minnesota, and I have had the privilege of working alongside Alice for 2001. And so about 2006, I became president of the board with her, and so we've been together for 18 years. And you'll know SADS Live is, I think, is it episode 155 next Friday? How many of you have been to win? Let's see who wins. How many of you have been to one SADS Live in one of them? 155, okay? And stay up if you watch 10 or more. 25 or more of the 155. Oh, Rachel, 50 year winner, chicken dinner, 50 or more. Who has watched all 155? I've watched them in real time. I haven't watched a single one, nor has my wife or children. I think they're like, what's Dad doing? I love doing those, but I hope you benefited from that. With that, and you'll, if you've done any of our SADS Lives, you'll probably say SADS Nation, or reason why the SADS Foundation is. You'll also know that I will, on behalf of Jan, our Director of Development, tell you guys that if you go to the website, www.stopsads.org, what's in the top right corner of the website? Remember, donate now. That red rectangle. Yeah. And so you can hit that donate button. And part of fueling the mission. And we need the fuel for the mission. And I was telling one of our colleagues in our program at Mayo Clinic, as I try to raise money for our research program, that one of the Sisters of St. Mary's, she's a famous nun at Mayo Clinic, and she would always say, no money, no mission, more money, more mission. And we can scale up our program accordingly to fulfill our mission. And you know, we have 4 pillars in our mission of SADS family: advocacy, family support, education, awareness. We are not a grant funding organization because there's just, that's not where we can put value at this time, at this scale of our program. You heard me mention before, we're about a $600,000 a year operating budget organization, and it's been about that for the last several years. And Jan, our Director of Development, has done an amazing job partnering with Alice to continue to sustain us in that way. But ultimately, we would like to grow because more money, more mission. So if you want to be part of that growth or the sustenance, here's the card right next to you guys. It is the Sustainer of Hearts pledge. And she tells me, Jan tells me, and I don't know if I should feel guilty by this comparison because I did grab this on the way over here this morning. But everyone at this conference gets a cost of a latte each month. Was it a venti or a grande or a tall? Okay, well, it does, because the grande was $6.38. Yeah, but guess what? Chestnut praline latte is out now. It's my favorite. I'm just really happy. But I did enjoy pumpkin spice, but I did a lot of pumpkin spice every day at Caribou, because our Starbucks closed in Mayo Clinic's basement. Terrible. But I moved on from the chestnut praline. So the cost of a chestnut praline by all of us would provide $1,000 a month of support from this room. So if you want to do it in that way, do it. If that feels like $6 a month from each of us, man, Jan, you undershot. How about a case? You know. Okay. So you can do this. You can fill out one of these cards. You can do the QR code. It's really fast. They've made it really easy to give. Imagine that. Really easy to give. So do the sustainer of the month. So, and join us in that way. That would be wonderful. Now, before we kick off the fireside chat, just in case we run up against the clock, I thought we would change the order, because I have to dash at 12:45 to catch the Uber. I think I told you that. So we're going to recognize the most important people, and that's the volunteer award. And so, Marshall, could you come up here and help announce, or Jan, the volunteer, and we'll recognize these incredible awards. We'll take the time for that, and then we'll shift into our conversation together. So first up, take it over.
All righty. We did have slides for this. We did. I had put them in the folder. I'm so sorry weren't found. Okay, so I will just go ahead and take over. We have very, very special people who volunteer for us every year, and we recognize a few of them every year. And this year we wanted to recognize a father and daughter duo, Aaron and Adeline Dinan. And if you've seen a little girl running around being interviewed, she was the Children's Miracle Network Champion of the Year. She had her little picture on the Wheaties box. She holds, I don't see them. Oh, okay, all right, there's Erin. She had told her story about long QT. It went viral. Then, with her dad's supervision, she started doing some videos for us on Instagram, and they also went viral. It was our first videos to go viral. Oh, perfect. So we wanted to recognize them as two of the volunteers of the year this year. So thank you so much. Oh, I'm jealous.
So thank you very much. And it was the most pressured interview I've ever had. Her daughter interviewed me this morning. I barely survived that interview, and so she's under the care of Dr. Andrew Landstrom, who, fun fact, is one of my trainees who's doing an amazing job. So the next generation is doing well. Oh.
You are one of our volunteers of the year, and you are just so awesome at spreading awareness about SADS. Thank you.
She did say I was her most favorite interview ever, right, Adeline? Right? Nod.
Alrighty, so the next volunteer of the year is Tim Hansen. He had a heart condition, originally thought it might be one of our conditions, and he called us up and thought Genevieve was one of the most special people ever, and he's right, she is. And he was so thrilled at the care that he got, but he ended up not having one of our conditions. But he was so pleased with the care he got and the support he got from us. He started volunteering with us. He now works with me every month on the finance committee. one of my favorite committees. Thank him so much.
Yeah, it was a journey for me last year. Being in my 60s and being diagnosed originally as ARVC, it was very unusual. And Genevieve and the SADS staff helped direct me, got me to Dr. Calkins and Brittany. And, you know, I went to that appointment that had my genetic testing, talked to the family, whole thing. And then one of the best things that he said, one of the saddest things he said to me was, "You're out of the club." You would think that would have been a great relief, but this is, my wife goes, "Really? Aren't you happy?" And I go, "Yeah, but there's such a great group here. So can I be part?" And that's why I volunteered. And I would tell you that the people here have just been so generous with their time, with their effort, that if you'd like to donate, please do. It really does do a great thing for people. I'm so happy here. I'm glad if you like to volunteer, I'd be glad to talk to you about how to do that. Thank you.
I don't know about you guys, but I'm blown away by that for Tim. When I think of how busy life is and busy time is, we generally give our time to things that are related to us, that advance our needs, our self-interest. And so to volunteer for an organization for the set of conditions that have nothing to do with Tim's existence or sphere for him, his children, his grandchildren, that's amazing stuff. So thank you for doing that. I didn't know that. So.
One last word, and then we're going to let Mike answer questions. Christy came to us through ARVC. She's one of our latest families to join us. And when she joined us. She's on the ARVC steering committee. She's a board member. She helps with other foundations. She is one of those people who found out and jumped in and started helping everywhere, and we're so glad she's with us.
Let's give a big hand to all of the volunteers of the year. And now the balance of time is yours. So had great presentations throughout the day, but we're bringing SADS live. So there's no question that's inappropriate. There's no question that's a stretch. And like I always tell those who've watched SADS live, I take your questions in real time. I don't cherry pick them. And so for now, I really will be hold to that. So we'll take them in real time. No, we'll skip that one. No, I'm just kidding.
I've heard some great things about gene therapy and also your involvement in supporting that. We haven't heard too much about the negatives or the risks, or like, should we be worried as parents for our kids, or is there something that can go wrong that we should be concerned about?
Yeah, so the question, if you didn't hear over here, is what's the state of gene therapy, and a lot of promise or hope, and could there be a little too much hype and not a balance on the risks? So I would say the risks to children are zero right now because we're not going to be enrolling children. So these gene therapies aren't getting launched for these SADS conditions. There are gene therapies that are underway, and we don't have to think too long to remember that there was a tragic death in the early stages of gene therapy in a 16-year-old, I believe, in Pennsylvania. So this is not a risk-free proposition. Nobody should think that all of the problems have been solved and that all we have to do is to get these patients enrolled and lickety-split, yellow brick road, here we are. But it's
super exciting to see the phase that we're at. I mean, we never talked about gene therapy as clinicians. We never mentioned it to you. I bet you during the first, for anybody who is 3 years or past when you were diagnosed, you had no discussion, no mention of gene therapy during your doctor visit. Not even on the radar. That's all changed, hasn't it? How many of you had heard the mention, if you asked what's upcoming during any of your doctor visits, is gene therapy on the horizon? Might it be possible? Now, those of us in the room, Dr. Webster, Dr. Landstrom, Dr. Barul, Dr. Chandler, we're saying, well, you know what? There's some problem. We're actually enrolling patients. And so one of our supporters, Tenaya Therapeutics, another supporter, Rocket Pharma, third supporter, Lexio, all three of them have human trials of gene therapy for PKP2 ARVC. That's never before happened. That is like right now, and those patients are being enrolled. Now, enrollment is admittedly super slow, and we're in the very early innings of this. The trials are really those who have enrolled, and I don't know if anybody's enrolled in this room, they're the heroes, because when you enroll in a trial that's what's called a phase 1, you have to enroll expecting 0 chance at efficacy. They're not designed to prove efficacy, that they work, that your disease gets better. They're designed solely to answer what you just asked, safety, and to continue to mount evidence that this is safe. And the news, and it's not just these trials, but the collective body of AAV-based gene therapy trials across all diseases has been really encouraging on the safety signal. There's still some things that concern us about the immune response to seeing the adeno-associated virus and so forth, but the safety signal is really encouraging at this time. If that safety signal is officially met with these early trials, then they will move on, assuming that these companies have not run out of runway. These are biopharma companies you mentioned, I know you asked yesterday, why does Thrive Therapeutics trial, why will that cost something like $59 million? And that's a drug trial. Gene therapy trials are next level at a 0 more expensive. So assuming they haven't burned through their runway, and they can launch the next phase of the trial, then we'll be moving into what's called the IND-enabling study. That is the safety and efficacy trial for which they would use that body of evidence to submit to the FDA for approval. That's why I told you 5 years. And I know the CEO of Thrive said, oh, yeah, we'll hit 5 years. Just trust me. Set your clock at 5 years. It's not going to be sooner. If we have any new therapy approved sooner than 5 years, that will be awesome. That will exceed expectations. But I hope that will help reset your calibration. I know Andrew Landstrom, he's given, how many of you have watched any of Andrew's webinars on gene therapy for the SADS Foundation? I know he's done several. So one of the common questions is, how soon will it be here? And so if anybody's thinking gene therapy will be in practice, us injecting our patients sooner than 5 years, I would say recalibrate your expectation. You'll be more satisfied with the state of our progress when we nail it in 7 years than when you're at 2 years from now and saying, why isn't it here yet? Because it's not going to be here yet. Andrew, your best guess, do you think I'm 5 years? Are you, if we're Las Vegas, are we under or over? But safety so far so good, and I think that's really encouraging. Our own, what we're excited, I can't announce it quite yet, but some of you know the journey with the Mayo discovery of suppression replacement gene therapy. This is a Mayo unique patented approach. It's completely different than PKP2. There's certain diseases where all you have to do is replenish, boost up the level of plakophilin for PKP2 families. Or that will not work for LQT2 families. That will not work for LQT1 families. That will not work for
a variety of diseases where you need to get rid of the bad marker. You can't just give more. And so that's why we have this strategy, out with the old, in with the new. We had, and by Burning Runway, it shows you how this can ebb and flow. So that was, the Mayo IP was bought a few years ago by Pfizer. We danced together for a while. We were making great progress treating those LQT1 wabbits. And then Pfizer divorced us summer of 2022. Pfizer not only divorced us, they parted with their entire rare cardiology unit and said, we're not going to have a rare cardiology program. So they just shipped that off. They shipped that off to a new company, but not with, but only with stuff that was Pfizer exclusive. So then we've been working the last 2 years to see if there another corporate partner who wants to work with us to co-develop it. So I was hoping the relationship would be signed before this meeting so we could announce it, but they're in the final innings with the lawyers. So hopefully that will give new legs and new energy and new momentum. And the priority, just so you know, I can announce the priority, there will be 6 different target lesions, heart conditions, and the number 1 target that's going to be advanced is LQT2. That's the priority target, even though we've made more progress on LQT1. And the reason behind that decision is all to do with what's called unmet therapeutic need. So the word on the street from the investors is that we've got LQT1 nailed. We've got it problem solved, it's good, there's not really unmet need, so why are we putting this much new energy and resources to something that we have solved? I disagree, and I think we heard from the families who participated in that forum this summer, June 6th, right? Wasn't it June 6th? Or June something, that you spoke loudly to the FDA and said you're not satisfied, but that's too much of an energy barrier to overcome. And so we are putting LQT1 behind it, because if we can prove that the therapy works for LQT2, then the energy barrier to overcome for the things that come next lessen. And so I'm not giving up for the LQT1 families. I'm not giving up. The rabbit trial went great. It basically made the rabbit's QT interval like the rabbit without long QT. So we know we have a strategy that's therapeutically effective. We're now working on the IND-enabling rabbit trial to then give the reason to be able to move that to human clinical trial. But that, what I just said there, if we're in a human clinical trial in LQT2 for gene therapy in 3 years, I mean, the clinical trial, not even submitting, I'll be super pleased with our progress. So that's, hopefully that gives you a feel of the pace and the cadence of progress. I know that sounds slow, but it's actually pretty fast. So, but that's the pace. Hopefully that, yeah.
Had an event, should he be taking a beta blocker as he's aging? Does that change anything about his past never having an event?
Yeah, so for those who didn't hear, is if I own a genetic marker, but I don't show it, in this case an LQT2 marker, and the doctors say I'm not showing it, I've never had anything, do I need to be on a prophylactic therapy anyway? In the case of long QT, that therapy is a beta blocker. That answer is it depends. I don't really care on what the age is of the patient. I don't care how long they've been asymptomatic if I don't like what the objective evidence is. But in a man with long QT syndrome who's reached that much age with that many years in the rearview mirror, they are trying to tell us, and they'll eventually teach us, what we will someday be able to call non-penetrant LQT2 or non-penetrant ARVC, meaning you own the marker, but it's never, ever, ever going to show itself. Right now, we don't have the knowledge or the skill to declare somebody the non-penetrant one, okay? But we know who is at super low risk, and there are certain individuals who are at super low risk.
So much so that we've actually started a program at Mayo where we do that, and we call it intentional non-therapy, where we know you own the marker, but we are consciously, intentionally choosing not to treat you every day with a prophylactic therapy. So we have over 130 patients now where we are choosing not to treat. And so far, 130 patients, about 10 years into this strategy, there's only been one patient, one of those 130, who had a self-limiting faint that made us say, okay, now we'll shift that person over to intentional therapy. So we know that we're over-treating. There's no doubt that we are over-treating you all, the community of genetic heart disease patients. And we're starting to learn which ones can we consciously undertreat, not just undertreat, not treat. And LQT men discovered in manhood after the age of 25, if they haven't had an event naturally yet, their likelihood of a subsequent event is super low. So if that fits then with their ECG is impressively normal, all of that goes to say they're at really low risk. But that doesn't, so non-therapy doesn't mean no therapy. So we call it intentional non-treatment with preventative measures only. So the number one way you can take an ultra-low-risk person from ultra-low risk to tragedy is not have told them about the QT drug hit list for our long QT families, and to have them get a QT drug exposure without our permission. So I have lots of patients who are on drugs on that hit list. That's different than somebody taking a patient that we don't know about and willy-nilly throwing them on Zithromax when they threw up 3 times the day before and their potassium is low. So that's the recommendation for somebody like low risk, like your husband, is the QT preventative measures. And there's lots of patients now where we're choosing not to treat their substrate. Yeah, Ms. Cook.
You mentioned the trials and awesome things coming from some of our sponsors here, specifically for type 1 and type 2 long QT syndrome. Our family is type 3, our daughter, and I'd love to know which one of these you might project would be aiming towards type 3 that you might put your hope into in the near future.
Yeah, so great question. So right now, none of the trials of the sponsors are LQT1 or LQT2. The one, well, I take that back. So the one trial from the sponsor that's in motion is Thrive Therapeutics, and that's where you met Mr. Truex yesterday. So Thrive Therapeutics on the right, they have a drug. It's called LQT1213. That's the name that they gave it. It's what's called an SGK1 inhibitor. Full disclosure, I've helped co-develop that drug. Mayo Clinic has relationship and so forth. But that drug has impressive QT shortening in the dish. It has impressive QT shortening of those rabbits, the LQT2 rabbits, shortening in the pigs, and shortening in humans. So some of you, if you want to raise your hand, we completed the first in-human trial of the drug in about 12 patients with LQT2, thank you, at the Spalding Clinic. And my patients who did that said that clinic was like a spa. It was a great experience, that they really did a great job of taking care of you and doing that. And the QT signal was really encouraging. And so now they're building the clinical trial. The earliest that trial will go into phase 2, phase 3 human enrollment is probably summer 2025 or later. Okay? Part of that summer or later is what you mentioned yesterday, is that thing called money, to raise the runway to be able to execute a trial of that size. That trial is going to be LQT2 and LQT3. So the patients for that drug is, it looks like, at least the early evidence is that this may be a much more promising drug for LQT2 and 3, Whether it will shorten the QT interval in LQT1, I'm less convinced, and because of that, the priority population is LQT2 and LQT3. So far, LQT3 is not easily amenable to gene therapy. Part of it is the size of the therapeutic cargo, meaning the gene SCN5A is too big. We think we've solved that problem in the dish, and so we're working with a new strategy to increase the cargo size, therapeutic cargo. We have a paper we'll be presenting and soon published showing that in the dish in humans with their LQT3, we can deliver the therapy and shorten the QT in the dish. But there's going to be a challenge with that, and the challenge is numbers. These companies need to have enough of the patients with that disease to actually get it ever into the macroeconomics to justify. So LQT2 being the most common, LQT1 and LQT2 are tied for first place for most common, depending on the study population. But for every 10 LQT2 families that might be in this room, there might be just 1 or 2 LQT3. So the numbers phenomenon starts to make LQT3 gene therapy less appealing, and so it's taken it's in the queue in the R&D pipeline, but pretty distant in the back. But the drug, this drug, and another drug that we've developed in my lab looks really encouraging for LQT3. So I think, but when we say we have a drug in the lab in the dish, that 5-year timetable that I gave you, might as well add another 5 years to your projection. So if it's in a trial like Thrive Therapeutics, I think 5 years is reasonable when you have it at that state of development. When we have something in the lab, we're trying to find a partner who will help us co-develop said compound, you might as well make that 5 plus 5 in terms of your timetable. And if we exceed that, that's bonus points. Let's go right here. Yeah.
So my son and I are both LQT1, and we found out in utero that he was LQT1. And so I'm curious, having heard you said that you medicate, treat, I forget what you said. Like, do you ever imagine a time, especially for our youngest patients, that they'll be free of beta blockers?
So, absolutely. Absolutely. We have lots of patients on beta blocker-free treatment strategies. So it will all depend on, and I don't know if you've heard talks on how do we decide what therapy do we give, how much do we give, when do we give. So we've moved from, not too long ago, the law of the land was thou shalt treat. Everybody gets treated, and everybody gets disqualified from everything. That's just circa 20 years ago, and circa 10 years ago, and circa last week for one of the families I just met. So there's still incredible disqualification posture that's out there. There's significant over-treatment posture that's out there. And a lot of it depends on who's taking care of them and what's their comfort level of going lighter on therapy. And so the less you understand the disease, you'll do one of two things out there in general. You'll either miss the disease, or you'll get the disease, but you'll overtreat the disease. And that's a real tendency that happens out there with these diagnoses. The more and more comfortable you get with the nature of the beast, you say, I think we can slay this beast without the sledgehammer. And you learn to try to get not too hot, not too cold, just right. And then what drives your movement to not too cold, not too hot, just right is how are we doing with said therapy? So if I have a family on beta blocker and he or she, your son, is thriving, I'm not that motivated to change course because we're thriving. If on the other hand, I have a patient where we say, you know, we traded my asymptomatic, dormant, never a symptom substrate for a new problem called every single day I feel lousy. Well, then the doctor can't say and shouldn't say, sorry, Charlie, this is how we're doing it. And so it's really working with the family to get it just right. And so we have families now, the 130 patients who are on non-treatment, you can imagine what they once were. They once were the beta-blocked ones who were the self-declared, what you guys use the term, I just have stolen it. It's called the beta-blocker zombie, okay? So whoever was the first to use that, it captures it. Now, it's given the false impression that everybody turns into a zombie on beta blockers, and that's just not true. So most patients tolerate beta blockers very well if you do it the right way. So if you take an asymptomatic patient and the next day you say, here's your 80 milligrams of Nadolol, you can become a guaranteed zombie. And so if you come upon the disease in an incidental way, there's no reason to take that incidental disease and go to stat zombie status, right? So if you go really slow, you can get it to where a lot of patients are at the dose that we would want them at, and they're saying, I like it, or at least I don't hate it. So those who hate it became the non-treated ones where we thought they could. Those who hated their beta blocker, but I said, look, I can't move to non-treatment because the risk score is there. So those have become our denervation solo therapy group. So we have about 120 patients who are one-time treated, called their denervation surgery now. So it's trying to find what's the right tool. So this week, during this meeting, before I came here, we just saw our 2,000th unique patient with long QT syndrome at Mayo Clinic. Of those 2,000, 100 are on non-therapy. That's only 5%. About that, 100 are on denervation one-time therapy. That's only 5%. So these unique ones, these are sort of tailor-made and being driven by how are we doing with the treatment strategy. But no, my goal is I will hope that when I retire next year, that we have moved to a beta blocker free strategy, and we'll be looking like beta blockers. That's so yesterday. I mean, that's how we did it in the past, but we have too many new options now that instead of being the first therapy, and it is, for long QT, it's the first therapy, that we would say, yeah, I don't think we even need to start with beta blockers. And maybe, and Dr. Chandler gave a talk yesterday on denervation solo therapy, we can't quite do denervation solo therapy as first therapy because then you'll get stuck with the bill. That's the only reason. Because the guidelines say denervation therapy is class 1, meaning that's what you do if your disease misbehaves and we need to do more, or your disease hasn't misbehaved but you hate your medicine, beta blocker side effects. Then the guidelines give permission to do the denervation, for which the insurance companies read that and say, okay, I guess we pay for it. That's the only reason in some of our patients we're not offering upfront denervation, one-time therapy be done. And I still think it's probably okay to do it that way. And so we do the trial of beta blocker. And for those families who are highly motivated to never take a beta blocker, we'll say, let's check in with each other to see if you're heading towards zombie status. And guess what? They all are when you set it up that way. But at least that way we can then document and say, we tried the standard of care, patient has unacceptable beta blocker side effects, now we're moving them to denervation one-time therapy or denervation lower dose therapy. And ultimately in those who do denervation with lower dose strategy, sometimes we take that lower dose number all the way to 0. So it's really, I mean, we have so many more treatment options than what we used to have for long QT syndrome. Great question. I want to make sure we aren't missing any over on. We'll do these over here, and then we'll turn over to the other side of the room. And let's go right here, Marcy. This gentleman.
So, question for you is, So what can we do to speed up the process? We, the average ordinary people, or people who might have something to it, some sort of condition. You had said that it's difficult to get people through the process or into certain programs for gene therapy. So I'm curious, what can we do about it? How do we get the whole process moving forward a little faster?
Well, thanks for that question. Overall, I think the pace, and you guys jump in too if you think otherwise, I think the pace is moving pretty swiftly. I mean, I think there's a healthiness to this methodical cadence, that we don't go too fast and then get snake bit. So for example, and Andrew knows this, in the gene therapy trials, there's a lot of competition, which is great. There's a PKP2 world in ARVC is crowded. 3 competitors. You might say, oh no, how can that work? Well, that probably is going to work ultimately, because now you have 3 groups of very smart teams exploring this. And while they want to win and get market share, they're not competing on safety. In other words, all of these companies have agreed to share best practices with the safety signal. And if they're seeing something and maybe we should be pre-treating with this, that, or the other, that's being shared. And that's really good for the progress. I don't think we should, like SADS, I think SADS is doing a great job to inform you all that there's a trial available. You can look into it. You can enroll if you want to. I don't twist arms of any of my patients to enroll, because enrolling in trials is complex. Those who went to Spalding, that was a commitment. That was a time commitment to do that. Not everybody has that time. Those who are enrolling in the PKP2 gene therapy trials, where there's 0 guarantee of efficacy and only safety, to me, that is flat. That's like, Tim, you volunteering for an entity that you have no disease. You have to be willing to enroll in a trial like that for the sake of the disease future community. Whether that disease future community is your children or grandchildren or people completely unrelated to you. They're the trailblazers. So I get it. I would have, for me, if I had it, I said, I'm not sure I want to be the learning exercise yet. And so everybody's willingness to be in that learning exercise, some people refer to it, I don't like the term, but guinea pig. So I just think we should be about informing, not making it hard to find out what trials are out there and what's the state of the trials and who can enter and how's the progress. We should, at SADS, do our best to make that information very available to you. But then let you all figure out, is it right for me to do this? Is it the right time of life? Is it the right situation? And so forth. And those who do the trials, all we can say is thank you, because we'll get nowhere without study subjects to figure out if any of this is working. The other part is $57 million. So if anybody is sitting on that, just give Thrive Therapeutics a call and say, hey, I got my friend at BlackRock, or whatever. But yeah, it just, when we're asking for a latte a month kind of territory to sustain SADS. I mean, drug development world, those people are tuned differently at the prospects of money burn and things like that. So if you happen to be connected, apparently I'm told that we're 1, 2, or 3 nodes away from serious wealth. I don't think that's true in my life. I'm somehow that expression that we're only a couple of contact points away from serious wealth, I'm not sure. But if that's true in your life, call your friend and first give to SADS, right, Jen? Then to one of these clinical trial organizations. Now, speaking of trials, I just mentioned CPVT families. It's been a bit of a bust so far. The Armgo Pharma trial is closed. So we were the lead enrollment site for the United States, Arthur Wilda for the United States. And by lead enrollment site, the only enrollment site, 2 patients. They've shut down that trial. So that potential new is no longer there, at least now. They're continuing that drug for a different disease, and if it advances for a different disease state, then we may have the drug available in the future off-label. And so right now, I think there's only one clinical trial available, and that's the Cardurian trial. And they similar to Armgo Pharma, are having a difficult time with study enrollment. Solid Biosciences, one of the givers here, they have a gene therapy trial that they're building the trial design around for CPVT, and it's using a unique strategy of delivering a protein called calsequestrin, and that counters the leaky problem. That company and that trial is going to have the same challenge that the drug trial companies have had, and that's study design. So study design right now, and I guess it's a good problem. Good problem is, because of flecainide, we hardly have any study-eligible subjects. So for our CPVT trials, you have to have enough residual ventricular ectopy to enroll in a study that shows said intervention got rid of the ventricular irritability. And when most of us, it used to be that when we would treat a patient with Nadolol and flecainide, we would look at your stress test, and if we had PVCs in by Gemini or we had a couplet, we'd say, yeah, we made incredible progress. Non-sustained VT's gone, triplets are gone, we just have by Gemini. And we'd say, good enough. Well, most of the CPVT centers are not saying good enough anymore with that. They're saying, let's have as our goal, with the current therapies that we do have, to cure your treadmill, to make your treadmill look like mine, with zero ectopy. That goal has resulted in the fact that there's no study subjects. So the companies have a challenge. They actually need to either hook up with physicians who have CPVT patients who are treating them the way we used to treat them 10 years ago, so that they have enough ectopy to enroll, or we're going to have to come up with a very clever study design where they'll be willing to get admitted to withdraw therapy to show the ectopy comes back, to put on therapy, and that is going to take a next level family of time to be in a CRU, a clinical trial study, for probably a, 10 to 14 day potential hospitalization. So we're meeting for the CPVT gene therapy trial in January to design the trial. And we all will learn, and I'll learn too, what is the official trial design, and are they going to be willing to, I think it's the only way we can do the study. I think the lessons learned by Cardurian and Armgo Pharma is, if we try to just find some patients where Andrew and Charlie and I and Dr. Chandler, where we said, yeah, we'll just leave you with the ectopy you have, so now that you have enough ectopy to enroll, they're just not, they're going to have a really hard time finding patients. So we're a victim of our success for CPVT. Flecainide has been an absolute game changer therapy for our CPVT families. And that's been the great, but that discovery that is now fully into practice, that has resulted in a real problem for patient enrollment. That was a great one. Anybody, I know this side had more of the questions. Any staying over here, working our way? Right here, Steve.
Just wondering, back to the beta blocker zombie topic, do you see anybody doing nutritional studies that are targeted to some of these drugs to see? And it's easy to find general nutritional guidelines, but is there anything out there, anybody working on, if you take this particular drug, maybe you're going to lose these nutrients and maybe this would be a better diet for you?
Yeah, that's a good question. So if you didn't hear, it's sort of nutrient studies, and is there any chance that our therapies are causing nutrient depletion in any tangible way? I'm not exactly sure of any direct studies. I can just say from clinical experience, I have not seen any signal of nutrient deficiencies with beta blocker therapy. Beta blockers have been used forever. And so that would be a really unusual one unless the child has an independent reason, and there's no channelopathy reason for nutrient issues. Now, having said that, that doesn't mean some of these channelopathies can't have non-cardiac organ signals. So LQT3 families, for example, sodium channel families, the sodium channel is a critical channel in the pacemaker of the gut, so GI motility. And so some families, if they've been dealing with gut dysmotility issues, I'm not ready to completely blow that off in an LQT3 or a Brugada family and say, oh, that has nothing to do with your genetic heart condition. It could. That answer, it could, doesn't practically change anything. But that just makes you realize that when we use the drug called mixilotine, and mixilotine is targeting and manipulating the sodium channel, that there's a reason, perhaps, why the number one side effect of mixilotine is sort of GI distress. And so it's, again, we have to dial in these medicines in a just right way. But I don't think from a micronutrient issue that either the disease or our therapies have anything to do with sort of micronutrient deficiencies. I just haven't seen that. Has anybody seen that? Yeah. Yes.
Finally. I am very curious, especially with the risk in long QT2 postpartum, if you know anything about hormonal birth controls and have recommendations on that for patients.
Great question. So LQT2 and postpartum, and that message, even one of the teenagers with LQT2, wonderful person who asked me the question over there in the teenage room, how many of you know that the word on the street is that LQT2 and postpartum is spooky? Yeah. So guess what? I'm going to tell you about that. Fake news. Okay? And the reason it's fake news is as follows. If you were to say, is postpartum an at-risk window of time for somebody who has undiagnosed and therefore untreated LQT2, correct answer is absolutely. Does it remain a high-risk window of time for women with diagnosed and treated LQT2? Almost never. So this is new data that will be presented next week by our team at American Heart here in Chicago that shows what's the rate of events postpartum in women with LQT2 diagnosed and treated. And the rate of events is almost zero. So what might be correctly labeled high risk, and I've never liked the term high risk, postpartum, the data was postpartum is higher risk than the 40 weeks of pregnancy or the 40 weeks before conception. That data was irrefutable. But does that postpartum high risk tag remain after diagnosis and treatment? No. So I think we're going to have more good news coming out again that we can debunk the notion for, I can't tell you how many LQT2 moms have concluded that they're not going to become moms because they say it's too spooky, the postpartum phase. I don't fear the postpartum phase in my LQT2 women at all. Now, do I look at that window and say, okay, how have we been treating you up till now? Have we made adjustments in the therapy as we enter that relatively higher risk window of time? Yeah, sometimes we make adjustments. Sometimes we put LQT2 women who were on beta blocker now on beta blocker and mixilotine for the first 9 months postpartum. And then we wean back off to their preconception state of therapy. So it's not as if we just don't do anything, but when we tailor the therapies the way we've been tailoring the therapies in our program, the postpartum event rate in diagnosed and treated LQT2 women is super low. So it's not spooky and high anymore. So I think that's going to be really encouraging when that message gets out. Marcia.
I have a question from the Zoom audience.
Yeah, and we love our Zoom. How many Zoom audience do we have?
I haven't heard today. Don't know the number. We'll find out. Yesterday we had 55
joined
plus
us online yesterday and today. So for the 17, it would be great to see you live with us, but I'm glad you joined us.
So the question is, will there be any gene therapy for Brugada syndrome ever? Wondered because it's supposedly so rare.
So Brugada syndrome and gene therapy, that's going to be problematic on several levels because the vast majority of Brugada is not a single gene monogenetic disease. Only 20% is. So now if Brugada is a 1 in 10,000 person disease and 20% of it is genetically mediated, that means 1 in 50,000 people have SCN5A mediated Brugada syndrome. That population size really shrinks for being amenable to a gene therapy solution. Now, some of you may say, yeah, but aren't there rarer diseases where we're doing some gene therapy? And the counter answer is yes. I mean, there's a gene therapy trial for Dannon's disease, which is super rare. But that disease has a high rate of heading to transplantation. And so unmet need, if it's there, and really there, allows the patient size to shrink a lot. But if we have a disease that's not perceived or isn't unmet need disease like Brugada, we do a really good job of treating Brugada overall. Because honestly, they either need nothing or they need a defibrillator. And then they just can't do marijuana. So besides that, they get to just do their thing. So, and cocaine, we take that away too, I mean. So it's got a problem where it's a 1 in 50,000 disorder that we do a really good job treating, for which the one gene therapy we could do is SCN5A, which is too big for now, although we think we've solved the too big problem. So we can get it to work, but I don't think the math is going to work. Certainly not for the first ones. If gene therapy ushers in a new era of cardiac precision medicine and no safety issues are emerging with experience, then the price point of gene therapy will decrease, everything decreases, everything gets easier to usher in the next wave of the gene therapies. But those were a lot of ifs, and ifs that have to be satisfied. Great question from online. Christine.
Backing off of that last question, we're a Brugada family, and as you know, my children both have Brugada syndrome. So I was just wondering if you have any recent data on how many pediatric Brugada patients are coming forward or like being discovered through genetic testing.
Yeah.
Worldwide.
Yeah.
I know it's rare.
Yeah, I mean, if more are coming forward through certain incidental genetic testings, I'm not sure. I haven't seen an uptick in pediatric Brugada patients to my clinic. I don't know if you guys have seen it in your practices, per se, in the last 3 years of increased awareness. Not really. I mean, it's a 1 in 10,000 person disorder. So, and every once in a while, I mean, very rarely, we will see patients with a sodium channel loss of function variant discovered incidentally coming to us from the world of the genetic odyssey cases, where kids with ill-defined whatever get a whole genome or a whole exome, and there's a certain list of genes, 72 of them or 68 or whatever, that if there's a variant of concern in it, even if that variant of concern has nothing to do with why the clinician ordered it for neurodevelopmental delay or GI whatever, then we'll find a few patients who have a sodium channel Brugada variant that way. But other than that, I can't think of increased uptake. Any of you guys? Stephanie, any? Yeah, not really. The community, and it's a good thing. I mean, the community, it's not as if we're missing patients. It's the Brugada community is a 1 in 10,000 person community. Long QT syndrome is a 1 in 2,000 person community. So, and I actually think long QT is probably going to end up closer to a 1 in 1,000 person community, because there's a lot of low risk substrate that lives under the water that we're learning about. So we may be discovering more and more of long QT syndrome, but discovering more and more of long QT syndrome that we may not have even needed to know about. So as long as we don't overdo it on that ultra low risk group that we're learning about and give them the safety precautions, drugs to avoid and so forth, then I'm happy to discover them. If we're just discovering more and more people who we didn't really need to find, who the uninformed physician puts them on a whopping dose of Nadolol stat, and then we make them miserable, then discovering more and more of these people is not called progress to me. So, yes, sir. Yeah. Give you a heads up, we're at 45 minutes on the counter, and after this question, we're going to take a little fun break, not a break, but I'm going to invite my patient and dear friend and author, who I hope many of you have gotten her book, but she's going to do a reading from her book for you, a brief reading. We'll take, what, a minute of the read? And so we'll have her come up, and then we'll continue with part 2 of our conversation. I suspect we could go on for longer than my flight, but I don't think I can reschedule. But so
like everyone, I'm excited by the prospects of the new medications and the gene therapies, but I'm also a little concerned that the insurance companies may be a barrier. They're ready, but they won't cover them, or the price is going to be too high, and it won't be affordable when it comes. Is there anything that is that, am I realistic in that thought, first off? Is that a realistic concern?
yes, I think
The volume won't be there.
yes and no. I mean, I think there's other genetic lesions for which there's gene therapy trials that are super rare, SMA, spinal muscular atrophy, where some of these therapies are unbelievably expensive, and they're being treated and paid for. So the precedent is, and you can be sure that a company making these drugs, along the way, as they're satisfying certain markers of success, they're readying the reimbursement landscape, because they're not going to do it. I mean, they're really, really good. They're not just advancing therapies and then say, well, we have no idea if it's going to get paid for. So they're doing their due diligence at the appropriate milestones. Now, are they talking to them now when we're still more than 1 year away? No. But do these companies know how to talk to the reimbursement world and get that readied? Absolutely. And so I mean, I think of my patients, I can't think of almost any patient who got their denervation surgery, which is not a cheap surgery, denied. Probably the more recent challenge that we've had from a current therapy that's available to you all is the newest generation ICD. So the EV ICD has been more challenging to get that viewed as covered because it doesn't have a specific code yet. And so for those of us who have chosen that ICD thinking that that might be the best way, we have to just ready the patient, say, it's just not zipped and covered like it would have been had we chose a different ICD. But already that is starting to improve. So I'm not worried about that being the Achilles heel. I'm more worried about, ultimately safety. These things have to be safe. And these companies have to survive. So I think the prospects of one or more of these companies that are supporting us now, not Medtronic, but one of the other companies not being on the runway in 2 years or being under a different name is real. And Medtronic, they'll be okay. They won't be one of them. But those other ones, they live on a typical 2-year, 3-year venture capital financial runway, they call it, in terms of their burn rate, they call it. And so we may not have them as a supporter in 2 or 3 years. They have to have something successful in their pipeline to garner interest, not in us, but in the money world, to enable new inflow of cash. So when Mr. Truitt said he's out there raising money, that is what he's out there doing, is to trying to make the case for unmet need. And he and I had lunch together, and the number one hesitation pushback among venture funders for long QT therapies is the venture capitalists say, it doesn't seem like there's unmet therapeutic need. And they, the money backers, are not convinced that there's unmet need. And so until they see the answer to the unmet need question, they're not putting their chip. There's chips to put in, but they're not putting them in yet. And so that's been his, and he's not the only one. The other entities that are in this space, it's been that unmet need question. So they haven't either been debriefed of that FDA summary that many of you participated in, which was huge. So it was hugely helpful for them, hugely helpful for us, but it hasn't penetrated to that world yet, and it needs to for all of us to win. I'm going to bring up my dear friend and author. She's going to do a little light reading for you, just for a little break, and then we'll do the next round of 40 minutes that we have and change. I can run to my hotel, so I can push that to 10-2 if I need to. But I'm going to give you, how many of you got her book? How many of you started reading it? How many of you read it already? Oh, okay. So for those who didn't read it, she's going to give you a little spoiler alert, just for fun.
Dr. Ackerman wanted me to share with you how we met. This is what you want me to read, right?
Whichever one you wanted, either that one
or the one at Mayo?
I don't know.
Okay. Well, this is... Maybe I should read that later one. Jeez, what am I doing?
Out your card.
Fill out your card.
You like that, Jan? Did you like that? Did I do good with it? Yeah.
Just shows you Dr. Ackerman's good sense of humor. I arrived alone and too early to Dr. Ackerman's talk at the Fox, filling my plate with free tamales, standing by myself at a tall table in the foyer to eat. 2 women edged toward my table. "Do you mind if we join you?" one of them said. Then she looked at me again. "Oh," she said, "you have an essay in Best American Essays 2016, right?" "I do," I said, flushing, pleased and surprised. "My husband and I saw your reading the other night," she said. "It was great." "That's so lovely," I said. "Thanks for coming." The woman introduced herself as Bridget. Her husband, she told me, was a pediatric cardiologist, a professor of pediatrics at the university. He would be escorting Dr. Ackerman. Not long after, her husband, Brent, arrived at the table, tall, boyish, and brown-haired, with a balding Dr. Ackerman in tow. Brent, should I forward on to this next description? When it was time to settle into the auditorium, I wandered into one of the front rows. Dr. Ackerman, fiddling with his mic, called down to me. "Are you sure you don't want to come up here instead?" he said. "The stage is ready for you." I laughed, shook my head. Dr. Ackerman had a dorky playfulness to him. His earnest Midwesternness seemed, up until that point, to be his overriding feature. But as the lights went down and his slideshow went up, I could suddenly feel the force of his intelligence. As he spoke, it became clear that he was not simply an activist physician. He was systematically using his research team to fill gap after gap in what the world knew about sudden arrhythmia death syndromes, including long QT. With each study result came a more refined next question. And as the results accrued, so did our ability to predict how the different types of long QT would act, and thus to filter out, for each type, the treatment methods that weren't as effective. The man had published more than 400 articles. I could see that while any number of electrophysiologists might be good at their jobs in the abstract, he was one of the few driving the conversation around long QT, which meant it was other electrophysiologists' job to keep up with his research. And did they? We young adults with long QT formed a tiny percentage of cardiac patients compared to the masses of elderly heart failure patients who poured into cardiologists' offices. What was the likelihood that a given doctor had seen the latest findings from Dr. Ackerman and his team? What did it mean to be a good long QT doctor? See, I used it against you so I could praise you.
Thank you. I love her book. I've read that sentence over and over again. But that was really special to read that. Her book. My favorite. And there's other parts in her book where, as her cardiologist, it's a little steamy. So there's a couple sections where you say, "Oh, much information." But the book, if for any of you that have done it, and I've never read a book like it, honestly, that unpacks the "I'm worthy to receive an ICD," where this machine. Wow. So it's just never read a book like it. It's fascinating. So thank you. And she is special as well, I take great comfort with that. If I look anything like Sixtus, I'm like, yeah, I have arrived. Another one of my patients' fathers, and he's only like the most handsome guy I think I've ever met, right? Right? You won't disagree, will you? No. Okay. 32 minutes on our counter for now, and we'll pick up where we left off next Friday at SADS Live. I don't remember where we're at, but next question is, I'll give a brag. Her daughter, I met when she was a little child, and her daughter is resident pediatric resident, Dr. Lindsay. So, was it 5 or 6 years old when I met her? Maybe a future pediatric cardiologist. Dr. Lindsay.
Intern, yeah, she did a stint with you at Mayo too. But you are awesome, Dr. Ackerman. I think we all can agree upon that. Question for you. As we do get older, you say that tired factor from Nadolol is dividing the dose let's say someone's on 20 milligrams, 10 in the morning and 10 at night, helpful with that tiredness, or does it not matter?
It depends on what age we're talking about, and, so for example, if an adult is on 20 milligrams of Nadolol, from a treatment standpoint, we're getting close to being on nothing. So at that dose, all we may be getting is side effect out of the equation rather than therapeutic need intent. So if somebody comes to me with beta blocker side effects, and we're moved down to that dose, I start asking myself the question, is it time to make the dose 0? I don't usually break the Nadolol dose in my adults to morning and evening because compliance is a big disadvantage or advantage. So as you know, I like the beta blocker therapy in my LQT1 patients to be morning time. My LQT2 patients, it's nighttime. And that comes from the observation that in LQT1, the majority of the events, if they're to occur, happen from noon to midnight. So I want the highest levels to correspond. In LQT2, they probably happen from midnight to noon. And so I'm a once-a-day Nadolol at that age, not in childhood because the child's liver doesn't quite processes the Nadolol too efficiently, such that we need twice-a-day dosing with Nadolol until about age 8, 9, 10-ish. Before we'll shift them to once a day. But if I have an adult who's really saying, I'm tired, then we have to unpack that. I mean, I'm on 0 Nadolol and I'm tired. So tired is multifactorial, but if I think it's beta blocker to blame, then I start asking myself, is it time to make their dose 0? Yeah, so ARVC, it's interesting. So ARVC, the use of Nadolol and flecainide for ARVC is pretty new, as in, I would say, 3 years old knowledge, and maybe buttressed in a nice study from Wojciech Zaryba within the last year. So how we now think of treating ARVC, especially PKP2 ARVC, for the electrical part of that disease, where we really treat PKP2 ARVC like I treat CPVT, Nadolol and flecainide. So there it's using those medicines and titrating them, and I titrate them, and I think, Andrew, you do the same, to making their Holter as clean as possible, making their stress test as clean, normal looking as possible. Newer in the ARVC world is for PKP2 patients, or honestly, any patient where there's a clear adrenaline trigger to their episode. So adrenaline, exercise triggered, faint, whatever, stress test that looks exercise triggering. We've started to provide denervation therapy to our ARVC community as an anti-adrenaline maneuver for that. And so the numbers are too small, too early to say, how pleased or impressed I am with it, but I'm pretty pleased and pretty impressed. So I think the order of denervation's therapeutic efficacy is going to be, in terms of which lesions does it work absolutely best for, it's going to be number 1, LQT1, number 2, CPVT, almost tied for first place, number 3, anything adrenaline triggering. Adrenaline-triggered HCM patients, adrenaline-triggered ARVC patients. Number last, LQT3. And I know that's where my dear friend Peter Schwartz and I differ. He thinks it's very effective in LQT3. I've been less impressed overall with its efficacy, but it could be different patients. So the LQT3 patients that we've denervated have been very early, very aggressive, very malignant, infantile LQT3. And I think for them, none of our current therapies are great enough. And so if they're doing mostly LQT3 in asymptomatic LQT3s that don't like their beta blocker, then of course it's going to look like it's working, because a good number of them probably don't need any therapy. So Nadolol and flecainide, ARVC titrated to trying to cure the Holter. Oh, for the dosing, dosing I do Nadolol morning, and flecainide morning and evening. So flecainide must be given twice a day unless we've proven that they're an ultra-poor metabolizer of flecainide to where we can, and about 3, 4, 5% of us are ultra-poor metabolizers of flecainide. And we can use that to our advantage if that person is, and then just dose them once a day if they were to be. Online world.
I have an online question. What's the risk for consuming caffeine for people with arrhythmogenic cardiomyopathy, from Justin?
Yeah, from Justin, risk of consuming caffeine. 0 if it's chestnut praline latte from Starbucks, grande size or less. No, so normal caffeine, and by normal caffeine I mean 100 milligrams or less territory. I think the proarrhythmic potential is un poquito in every one of our SADS conditions. We recently published a paper of a series of anecdotal close calls, sudden cardiac arrest, or tragic deaths temporally related to consuming highly caffeinated energy drinks. So in our program for all of our SADS patients, and this is easy to advise since there's actually no true redeeming health benefits of Monster, Red Bull, 5-Hour Energy, and those have a lot. As you know, there's a lawsuit, I don't know if it's been settled, of an LQT1 college student who had diagnosed and treated LQT1. She was an American Heart Association volunteer, wonderful person. I knew her mom, didn't get to be her physician, who died after Panera Powerade drink. And Panera pulled, now I think you can't get Powerade, or if you can get it, it now boldly says a whopping amount of caffeine is present in this drink. So the lawsuit had to deal with, there was no, it wasn't made obvious just how caffeinated Powerade is in that particular drink. So I think normal caffeine consumption, 0 risk in any of our SADS conditions, but it's easy. The other exogenous exposure, that's easy to say avoid, even though our true evidence is weak. We have a paper that we just published showing similar deaths, tragedies in the temporal relationship to vaping. So we and others have a growing concern that vaping is going to be rearing its head in the susceptible person. If you're healthy, you can probably handle vaping. It's the same thing. If you're healthy, Monster's gonna not hurt you at all. But if you have sudden death susceptibility variants, this could be the way that it rears its head, just like postpartum is a way that an LQT2 person gets found, or a near drowning is the way that a CPVT person or an LQT1 person who wasn't diagnosed and wasn't treated gets discovered. So for some, vaping or Monster or Red Bull could be the triggering episode that causes what otherwise would have been a dormant substrate to rear its head. Yes.
Kind of a follow-up to the postpartum question where you mentioned, look, sometimes we'll alter the treatment during that period. Do you take that approach for like adolescent or premenopausal, also kind of proactively when a child, maybe especially a girl, is approaching one of those phases? Or do you, like, you know, would you proactively bring that into consideration, or is it more, gee, if something happens during that time, that's when we assess?
Yeah, that's such a good question. A lot of it is science. Some of it is art now of just, you know, how do I justify with any kind of convincing that there's a gut instinct that you start to develop as to whether you need to make a preemptive move. So we try in our program, it's easy to make a reactive move. Any of us can do something, I hope, when a patient calls and says, guess what, I just fainted during track. Oh, it looks like I better do more. That's like easy. It's sort of seeing something that's just bothering you a little bit to where you say, I think I need to nudge up this dose. And seeing it and feeling like you need to intervene before badness happens, which brings me to one of my favorite movies, Tom Cruise's Minority Report. Anybody seen that? Where He sees the ball that tells him where the murder is about to happen, and then he intercepts it just before the murder happens. Great movie. So I think sometimes we get this sense of, I think I need to do something. And so we do that in our women. Our girls, so I have a lot of girls with LQT2, for example, who from birth to 12, they're not treated. So we do not embrace the idea of mandatory treatment. So if I think they're super low risk and that girl with LQT2 is in a really low risk window of time, we don't say you still have to be on a beta blocker. But as we nudge towards the arrival of estrogen, which is QT twitchy, then we ask the question, is she changing? Is she becoming a reactor? So we'll do things like after your third menstrual cycle, I want you to get an ECG locally so that I can see your ECG in the setting of estrogen versus our first 12 years of life together. Has it changed? Is it changing? Is it reacting? And then I might up the ante and say, wow, you went from a very, very normal to a wow. Now, I must tell you, going from a very, very normal QT to a wow during estrogen's arrival is like almost never. It's been interesting to us now that we've been doing it this way. But we'll look to see, and then we'll say, I want to now do, if I've been watching that child, say, for example, every 3 years during childhood, on the arrival of estrogen, we're now at yearly check-in. So we're doing increased surveillance, looking for things that now we don't quite like. Some of them will now commit to therapy. We have boys that we've been on beta blocker therapy until testosterone's arrival, and then we start to wean them off of their beta blocker or stop doing the, you now weigh 60 kilos because you're a big teenager, so now let's rev up your natal law to 60. We'll say, wait a second, we now are in a fundamentally lower risk window of life. We don't need to just keep, the teaching used to be, you keep cranking up the natal law as you get bigger and bigger. We stopped cranking it up like 10 years ago in our program. And so all of these things we're of course, I mean, to me, preventing tragedy is easy in our program. So our goal is not preventing sudden death, because that's just honestly not hard. Our goal is life large. We want life large living kind of stuff, not just, well, nothing bad has happened, therefore we're doing great. I want nothing bad to happen and the family to tell me we're doing great. That's my definition of winning with these. And so, yes, we'll do that. Menopause, the transition to menopause, honestly, I think we have missed, I don't think menopause is creates much new risk in LQT2. I think that data has not been very convincing. I have not seen spookiness emerge. Now, oral contraception, I think somebody might have asked, I can't shout this from the mountaintop about which oral contraceptive am I concerned about, but we published a paper many years ago where we have concerns about Depo-Provera as the form. So that the progesterone-only formulation, we had several LQT2 women, and got me convinced enough. She got her Depo. A month later, she had her shock, removed her Depo, a year free of no shock. The next year, she tried the Depo again, got a shock. We're like, okay, that's pretty convincing. And then we did some studies in the dish and showed that Depo-Provera is a bit electrically irritating to the long QT cells in the dish. So to me, that's enough preliminary evidence, especially when there's other great oral contraceptive strategies besides the 3-month Depo injection. So that's the only form of hormonal contraception that I sort of discourage, I guess.
So I want to go back to the Nadolol. So you went from, it used to be like maybe a Tenolol, and then you went to Nadolol because it was longer acting. And you say give it, so for long QT1, you would give it in the morning. So if I take my dose, when does it start really working in your bloodstream? So I take it, takes me 30 minutes to get to the pool. Am I okay? And then if it is longer lasting, like how long does it stay in your system at high enough levels?
So great question. So first, we didn't go from atenolol to Nadolol because Nadolol was once a day. We went from atenolol to Nadolol because atenolol is worthless. So atenolol, if nobody ever took atenolol again, I'd be happy, at least for my SADS conditions. It's just not as good of a, it'll never be proved, what that statement I just said with confidence will never ever get proven in a randomized trial. Because nobody, no long QT specialist that I know of would ever randomize his or her patients to exposure to atenolol, because we are that convinced with the retrospective experiential observation data. The Nadolol, no one should ever worry about their morning dose and when they could maybe do something if they're taking it. So when you're taking your Nadolol faithfully, the beauty of it, it's chemically long acting. So there's what's called a steady state, a residual level that builds. Now, why do I like morning for LQT1 and evening for LQT2? Maybe a little voodoo, but it makes sense that with any dose you give, there will be a transient peak spike and then returning back to that steady state level. Do we need to have the peak timed during the window of time like we do in LQT1, LQT2? Probably not. So to the point where if I have an LQT2 person who says, you know, I'm just missing the evening dose, not where I have a good habit, then I said, great, take it in the morning. Because that spike idea is more conceptual in my head, and I would say the enemy of good is better. So even though if I think it might be slightly better for LQT2 at night, what's better is taking it at a time that you basically never miss. That's better. And so if that's your better, then great. But a person doesn't need, an LQT1 person doesn't need to say, okay, I just took it at 8. I can't do basketball practice until 9:30 when the peak of the drug does its peak thing. So we really don't have to do timing, thinking about our activities of life in reference to when I swallowed the medication. That's a really good question. Yes, sir, Tim, our Volunteer of the Year awardee. Yeah.
I know that you don't necessarily recommend it for LQ, for the other one, but for us that are not that, is it?
Yeah. Metoprolol is a great beta blocker.
Okay.
It's just, it turns out that in long QT syndrome, the secret to Nadolol success and propranolol success, so both of those beta blockers I like a lot, is they're not just beta blockers. So those two drugs, Nadolol, propranolol, are also called what's called alpha blockers. And some of us think that it's the combination that gives its differential therapeutic efficacy over atenolol and metoprolol, which those two beta blockers are really what are called pure beta blockers. Now, metoprolol in a heart failure patient is the standard of care, the #1 beta blocker, basically. Metoprolol or carvedilol, Coreg. So a lot of ARVC families are on metoprolol. And if the purpose of the metoprolol is more mechanical function, then I love it a lot. If you were to ask me which beta blocker do I think is the best quote-unquote antiarrhythmic beta blocker, then I give props to Nadolol because the alpha antagonism is better. In fact, Nadolol probably most closely mimics, Nadolol and an IV drug called labetalol, probably most closely chemically mimics what left-sided denervation surgery is doing. And so as an antifibrillatory, antiarrhythmic, and I'm saying that loosely in terms of that, but that's why in an ARVC patient whose mechanical function is perfect, their squeeze is normal, but they have Holter ectopy, they have exercise stress test ectopy. In them, I like Nadolol and flecainide. If they already have pump dysfunction and they're going to need to be on a beta blocker for pump health, if you will, then I'm happy with the metoprolol and flecainide because I think in them, the flecainide is probably the better antiarrhythmic component to their treatment strategy. Yes.
our other Volunteer of the Year award winner and fellow board member.
And just a quick follow-up to that, not that I don't want to kill you on beta blockers. At the same time, what you were saying about metoprolol is ringing, with me because I am an ACM patient. But dizziness and side effects, is Nadolol easier on side effects than metoprolol succinate extended release or no?
Yeah, the side effect package for beta blockers, I would say, is really patient specific.
Okay.
And that's why I'm willing, in general, if somebody hated propranolol, they're going to hate Nadolol. If somebody hated Nadolol, they're going to hate propranolol. So we're willing, but if they say, I'm not ready for denervation, for example, in that set, if that's where we're heading, if they want to do a trial of a second beta blocker and say, I equally dislike this one, we do. We used to do that more. Now more and more families are like, no, I don't like this beta blocker, let's move to surgery. But we have had plenty of patients where one beta blocker disagreed with them a lot, and the other one they were satisfied with. Satisfied enough to say, for now, I'd rather stay on this strategy than embrace the surgery strategy. Surgeries are not free of complications. Right. Yep. Devices are not free. So if they get content and say, but now, all things considered, I'd rather stay on the medicine with this package that I know, I'm content. If you're content, then I'm content.
Not content.
Yeah, but yeah. So and for me, if you're not content, that drives everything, because I think we should be tailoring therapy with the goal towards contentment, not just residual. Well, I guess this is my lot, so be it. I just think we ought to be striving to do way better than that. Yeah.
Thanks.
Yeah, well, I think running is good for long QT. 5 more minutes max? Okay, great. I thought you were thinking me. I'm still good. I can fly to my hotel. No, yep. Nope. So the boss, and let's give a big hand for Marcia, who's done all of this. She's like the best, and she's put together an incredible 2 days for all of us. But the boss has given me the clock. Perfect. Well, then we should 1:15. Oh, yep.
Okay, just a quick question with the genetic therapies. Will they actually improve the condition or just hold it in check?
Yeah. So it depends on which disease we're thinking of. So the goal in long QT syndrome is not just to improve, it's to bring the QT interval to normal. That would be the goal. The goal of ARVC gene therapy, we don't know yet. So patient selection is going to be very important. So PKP2, ARVC, those are the 3 companies. We don't know the answer to when do we need to replenish? Or is there a certain stage in the disease that's a bridge too far that if you start replenishing the plakophilin, did we miss our window of opportunity for what we would call reverse remodeling? So in cardiac world, remodeling is not a good thing. In your bathroom, remodeling is what you're spending money on, right? But in cardiology, remodeling is a bad thing. That's what the heart is doing in reaction to the disease process. So our therapies, the goal is to reverse remodel. That's the correct word. Is there a window of opportunity that we have to strike? Meaning, do we need to give the gene therapy before the pump gets weaker, before we see too much scar on the MRI? And as you can imagine, we're not enrolling those patients in these initial trials because you tend to have to enroll sicker patients. So we may learn that there's the sweet spot in time for certain disease. For long QT, it's not going to matter. If you need to be treated, your QTC is over. So who will get the first gene therapy in long QT syndrome? It'll be long QT patients with a defibrillator with a QTC over 500. I can almost guarantee you that will be the study design. Because then we'll have safety covered should we need it. 500 so we can show that it works? Does it bring the 500 to 420? So that's the goal in the long QT therapy. In the PKP2 world of gene therapy, it's sort of to be determined when do we have to strike in terms of the therapeutic window. Ma'am, I'm going to give you the final one of the day.
Probably not a good question either. It's nutrition related. Okay. So my son has the PKP2 mutation, as does his son, who's one year old now, 1.5. And I'm worried about nutrition and the effect of nutrition on accelerating possible disease.
Yeah.
Because he's a vegan. They're feeding him as a vegan. And I worry about whether or not there's enough of the right mix of amino acid. Is he getting enough protein? And is that related at all to any of the disease patient or present?
Yeah, that's a great question. I would say in general, we have no data of dietary preferences being a disease accelerator, disease aggravator to any of our diseases. So I would say from that standpoint, I won't be worried. And so I'm a full-fledged carnivore, so sorry about that. So I would be more asking the pediatrician, the family medicine, are they content, satisfied with nutritional health with a vegan strategy at age 1.5 or 18 months? And if the pediatrician is saying, yeah, we can do this, and maybe I want you to take a Flintstone multivitamin or whatever, then fine. I would not feel that we would need to talk that son or grandson into a different dietary nutrition approach in the name of, I think this could be dangerous for your PKP2. I don't think I don't see any signal of concern with that strategy, although I would admit that the database to say I don't see any risk of concern, there's almost zero data. So it just would be an impression that I think if the family medicine doc, pediatrician said, no concern with vegan in an 18-month, and I don't know what that answer is. The pediatrics was a long window away from me to speak with any sort of confidence on what is the state. Would a pediatrician support veganism in an 18-month-old? I don't know that answer. But if they did, then I'd be okay with it from the standpoint of taking care of your grandson's heart. Well, I thought maybe that we would run out of gas 45 minutes ago, but you guys were amazing. This was a blast. So we're taking a break. Marcia says head to lunch and eat fast. And goodbye, take care, and remember, our whole purpose is to live and thrive. So I hope you spend your next day and years living and thriving. Until next time, thank you for everything, everybody. Blessings.
Thank you, Dr. Ackerman. All right.